going to in this session, as I told earlier, I don't know how much you people are aware about the microbial science and how each microbes are important for the human body, important for the playing roles in our life. And those are the newcomers, and those are learning biology and bioinformatics. They should basically know about how the things are going and how we are surrounding with the microbes. So microbes, in the sense, it includes your virus, fungi, and your bacteria. And bacteria may be pathogenic bacteria, maybe like probiotics is also that there are different bacteria. Those are harmful there, and some of the bacteria are also pathogenic actually. So pathogenic means harmful bacteria, and some of the probiotics, those are bacteria, though they are beneficial to our you can say the health actually. So after you can say the sequencing all those things, people started focusing on the microbial science because microbes are teeny means they are very less, and basically they are having the sequencing cost for them is very less, and they are having fast we can sequence it, and they are having wild varieties. There are like millions of varieties or species are available for the microbes. So we have touched only thousands or 2000 of the well-known sequencing or you can say assemblies of those microbes, but still multiple texts are blank in NCBI. If you can go into the NCBI and see microbial taxonomy, we have the names over there, but we do not find any sequences related to them, and there are multiple gaps are there actually. So now entire word like Pritish sir added here, we need to go for the Indian population's gut microbes, microbial identification. And apart from gut, people are taking different samples from the different part of the body, like skin microbial science is also there. And then there are multiple body parts are there where we are surrounded with the microbes, and they are playing important roles over there. Of course, the gut microbes are the big one, which are playing more role in terms of the different scenario and in terms of the regulation of our health. So because they are present in the digestion system or gut of the human, and they can play a lot of roles there. So your body will behave how they are. Now the people are seeing they are the deciding factor. And definitely a surrounding environment wherever you are living, what kind of the microbial community are present, and how you are behaving over there, and how your immunity, basically you are behaving means your immunity is behaving to harmful bacteria or accepting to the you can say the beneficial bacterias or pathogens or whatever. Beneficial pathogen will be not there, definitely bacteria are there, those named as a probiotics. I will take terms as a probiotics. And pathogen means the harmful bacteria, and then you can go with the viruses again the harmful, and how they are interacting and how this is. So here in this series, like I will not say series, this 1.5 hour or 2 hour session, I will try to provide you the overview of like how we are surrounded with this, and how our those microbes or microfilaments or those how they are interacting with our human cell, and how they are replicating their self inside the body, including viruses. I think those are latestly SARS-CoV virus. People have gone through a lot of literature to them, how they are replicating, how they are behaving inside the body, how the interaction point are there, attachment point are there. So that science is well known now, and that process is well known now in case of SARS-CoV virus. But other like fungi has the flagellum over there, and bacteria is also having some you can say the epitopes type of the things. They are interacting with the human cells, and what are the different our different receptors are there, and they are making to make inside get inside to the body, to the particular cell, and then they will start functioning over there. So they can take participation in terms of the positive way. Positive way means they can regulate they can regulate different metabolites, and they can form because microbial after getting inside the body, microbes also produce some kind of the you can say some kind of the metabolites. Those are beneficial to the human body, and they can work as a catalytic or maybe enhancer to the different genes and proteins. Those are helpful in certain disease, or make it, I can say the chronic disease, such as diabetes, cancer, inflammatory IBL. IBL is the most studied disease in human case. Inflammatory bowel disease are the most studied disease, and diabetes also it is playing very much role. And also because diabetes is directly related to your consumption of the foods actually, and what kind of the food you are eating, what kind of the bacteria are surviving there, and how it is happening. That kind of the biologies are going again related to that. So I just try to summarize in the figure. Like there is like a nice figure I got from one paper actually, and what kind of the different receptors are basically participating to receive those viruses or bacterial different filaments of the bacteria actually. So you can see the different filaments of the bacteria and their particular attachment, how they are attaching and to these cell receptors. So LDLR receptor is there, TLR4 receptor is there, IgG receptors is up there, and HP H of protein 91B, which is generally now called GP96 protein, is also there. So they are different section, and they are participating to interaction of that. So here I'm just putting the biology concept here, how they are interacting and interacting and making entry inside the host, and then making replication, their own replication, and millions of the bacteria will be grow over there, and then they will start producing the different kind of the metabolites over there. So like assembly and budding part will be there actually. So this is like entire process which is happening, and some of them are valley. I cannot say everything is clear now. No, there are like many things has to be decided, many interaction method in wet lab. It has to be more keen, fine tune methods should be still remaining, because that is that area, or you can say that microbiome, and when the genome sequencing has been come and the microbiome has started in the parallel way, but still people are not considering more. But yeah, nowadays the people or scientists across the world are focusing more. Those are studying human health system actually. So in that way it is happening, and this is the entire schemata, a visual schemata, how the microbes, different kind of microbes are. But yeah, it depends on the different. In case of bacteria, interaction mechanism will be different. In case of the virus, then again the different. Then fungi case, there again the things will be different. So I'm just put here the overall picture over there. And in the next session I will try to like how the things are evaluated, like the process of evaluating the host microbe. Because once we get the microbe inside, then how we will check whether the microbes are there, and how the technologies are helping us to find out those interactions, especially in terms of like you can say the proteomics and meta transcriptomic approach. And what are the different methods and technology those are helping to find out like a new species of the microbe, or you can say uh, mm, yeah, microbe as well as a strain over there. There are numbers of strains are there in case of the bacteria. So these are the few because these maximum these are the RNA things are there. So maximum sequencing technology are considering like 60. So 16S are like old technology, which are basically identifying the stretch of the viruses. Some known stretch are there, and then based on that stretch, whole bacteria are whole, not whole bacteria, but region of the bacteria are sequenced, and then we are capable to classify them into the different category of the bacteria or microbes. Again, V3, V4 is one of the similar kind of the technology, but these two technologies are like ITS is also the similar kind, but these three technology are the older kind of the technology when we are not having much super fast sequencing technologies with us. But now we have metatranscriptome and whole kind of the metatranscriptome means, because these are the RNA, maximum RNA containing virus are there, RNA we have bacteria and all those things. So metatranscriptome technology are participating to sequence entire bacteria, virus or fungi, and will get the in corresponding sequences with us. And then when we are coming to the analyzing part of the you can say host, with host how it is behaving, and host what are the different host proteins are participating with the interaction with microbes. Again, I will repeating the same thing. Microbes means bacteria, fungi, and all those things. How they are behaving. So there is a how we can find out those interaction in based on RNA technology. That is like we used to call that a dual RNA sequencing part, where we know which host we have collected that particular sample, whether it is human, whether it is like animal, any kind of the animal, or whether it is plant. So I think yesterday you have gone through RNA sequencing technology, which you are having a one single kind of the sequencing are collected from the same kind of the from a tissue, and we are processing with one reference genome or de novo. Okay, but when we are considering dual RNA sequencing technology, here we are considering two types of the genome. One is the host genome, another one is like microbe genome, microbial. You can say the taxonomy. So microbial taxonomy is defined again because it's have a lot of number of species are there. So we used to call, when you got the sequenced out from there, we used to call the OTUs form. So each OTUs belongs to the each type of the species. So this kind of the classifications are there, and they are coming out there. Once we get, and there is another methods is also there. Once we get out the proteomics data or metatranscriptome data out from there, we can go for the exo metatranscriptome analysis and host proteome analysis. Once we filter out the different proteins out from there, then we can go for the quantitative approach like HPLC, LCMS approach, and all other kind of the approach. Culture them in vivo and try to find out the their database related related parameter wise approaches from there, and then we can submit the public database is there, and then try to map them. And after mapping them, and we'll get the different kind of the interaction analysis and the data analysis part, if we are considering the proteomics and metaproteomics approach. But apart from that, we are also having you can say the science is growing and the people are working in the n number of the direction in that. We are also having the different computational method. Those are quite capable. I cannot say they will give you the 100% result, but yeah, quite capable to identify host microbe interaction. So like I think those are the biotechnology student, they will love that. I think they have gone through something like biochemical analysis or bioprocess analysis earlier in the BTech courses. One is method is called the methods interfering metabolomic interaction, to identify the metabolomic interaction with host. There is a method which we generally follow, how particular microbes are going inside the body in a certain environment, and how what kind of the different metabolites it is producing, and if it is producing X metabolites, how this particular X metabolites are participating to regulate the particular protein or gene inside the human body. So we called as genome-wide metabolic interactions. Actually, you can say genome scale mechanism of metabolic interaction network. We can consult on that. And then another method is like protein interaction method, where we are nowadays having different kind of the approaches are available. One is like machine learning based approaches are there, and another one is like structural feature based approaches are there, and then we will also having some literature mining based approaches are there, and then interlock tool based approaches also there. So each of these technology I am going to discuss in detail in further PPT, or further as the lecture are going to the process. And then we have RNA target domain prediction based approaches is also there. And one more, like you can say, specific metabolics intra-species or inter-species approaches is also there. That is basically depend on your experiment design, and what kind of the approaches we are using to identify the those things actually. I hope I have made somehow four methods which are using computationally. People are using to identify them. Not tougher, but yeah, people are taking like host microbe interaction using microbial mediated pathways actually. So
Yes.
just start like how this metabolite mediated pathways are computationally we can model them, and we can find out the relationship between the interaction with host. So there are like many tools are available. Those are use the genomic, metabolomics, and phenotypic information of the things, and they will try to model them. These are basically some of them like are based on the system biology with mathematical modeling and correlation with them. So like biopsych is a reference database which is having the all kind of the pathway modeling over there, and where we can find out the interaction. And then eCell is there, and pathway tool is also the one of the big player nowadays, where we are having all bacteria's defined pathways are defined, and especially for in case of E. coli and other kind of the bacteria there. One of the well-known MATLAB tool is Cobra, which is participating in this metabolite-mediated pathways. And this tool is also available. I have used this tool. This is very fantastic tool to get in a gut environment. If you have a gut environment, human body gut environment, we can set the gut environment across it's like a protein modeling or dynamics we are doing. Similar kind of the things we are doing here. So suppose we have a gut environment, we have a metabolic proteins, or we have a bacterial protein, or you can say different bacteria, Bacillus subtilis, E. coli, and other bacterial genomic information, and then we can able to predict what kind of the possible fluxes are coming from that. Fluxes means what kind of possible metabolites are coming if they are inside into the human gut environment. So basically I'm just trying to throw a picture in such a way where we are making a biological environment in a computer, and then we are having a specific virus or a specific bacteria is there, and then that particular bacteria how it is behaving, and how what kind of the substrate they are producing. Substrate means metabolites are producing. As I earlier told, these metabolites are very important to regulate the system of gut. So in this type of the analysis, we are going to we are going to create a such environment, and after that, in that environment we have to give the flux in and flux out also. So the bacteria is eating glucose from the environment, and they are basically in a picture you can see they are like, suppose I am giving one example of B adolescent and adolescent synthesis over there. So they are producing the acetate, which are important for some of the reactions actually, and then we are producing the fumarate, ethanol, and lactase also. So these kind of so some of the bacteria are also lactic acid bacteria is also there, which are important. In case of, you can say, in yeast bacteria, you can say in curd where there are yeast bacteria are there, they are important, and they also produce some biomass. So these kind of the tools will help to identify what kind of the end product are coming from that, and what kind of the biomass is also coming. So biomass composition is also different over there. So if biomass composition have their own category, and if biomass is that can be consumed by the cell or not, that is also important. So these kind of the things we have to find out, and find out the way and identify them. So if it is any, like, you can say that any new, this is if we have a well-known bacteria, a well-known environment over there, we can produce them. So this kind of the technology is basically using in the probiotic industry. Probiotic industry means those are like food producer, like there are different kind of the milkshake, different kind of the like curd they are making, different kind of the you can say flavonoid food they are making. So some of them is like, you heard about the yogurt name is there. So in yogurt, what kind of the different bacteria are there? So basically they have to scientifically design, if there are n number of bacteria are there, like suppose 10 bacteria are there, so the possible and if that yogurt food is going inside the body, then what kind of the metabolites are going to produce. So this kind of the system, like basically this metabolite mediated whatever analysis we are doing computationally will helpful to identify those actually. Coming to the, and I can go for that modeling. Modeling also the big task or important task here. Important means if you are missing, if you are not having the good optimization in the modeling, the result will be not as good as we are expecting over there. So and it is also computationally intensive actually. Sorry. So these are the different OptCam, Beck Arena, and DYMM, and MISO, and ComSat, and Flycon, and MOI. These are the like a specific model, a specific based on the species they have developed these kind of the tools, and we have the data over there. And I will focus into the K-base. K-base is the entire, you can say, a big database where the more focus has been given to the microbial science, and all kind of the microbial species are present, and all kind of the microbial analysis are present over there. So if you get the time, you will go in this particular database. I will also go a little bit of that particular database and how it is operating. Here I just put it a slide how we can create a you can say a metabolite network in a certain gut environment or any kind of the environment. This is like an overview. I don't know how depth you will get from this, but I will try to put in such a manner where we can you can get the information from here. Suppose we have a genomic strain, and we don't know whether it is belong to the known strain or whether it is belong to the unknown strain. So if we have a known strain, we know in E. coli also there are number of strains are there. Each E. coli strains will produce a certain kind, like maximum all of the strain are having producing the such one substrate or metabolite. Those are common, but some of them will produce high, some of them produce the content of metabolites will low. So in that way we can classify them. Then we have to go the like we have a strain genome of the strain. Once we get the genome of the strain, we'll go for the prediction of the metabolic pathway. Then we will that prediction of the metabolic pathway is quite tough task. There are multiple tools are involving in that. We will predict all the different metabolic pathway by based on, like you can say, the basic approach is like based on the, we have a sequences with us, we can go and align with the different metabolic databases. After aligning with the different metabolic databases, we will find a certain function of that particular gene, function of the particular protein, and then we'll have a set of pathways over there, whether that is fitting in the particular pathway or not. We will go for that actually, and then we go for the gap filling if some of the you can say some of the proteins are not identified, and that particular you can say not produced by that particular bacteria. We have to see whether it is not produced or we can fill the gap. For the gap filling we have to optimize. Then we can go for the model validation. Once we constructed the model, we have to validate that particular model also. And then again, yeah, after validating the model, we have to go the flex balance analysis. That is called like we are giving feeding to our model, and what kind of the metabolites are produced by these models. I will go for the some of the experiment based things actually over here, and we'll go. But then after flex balance analysis, flex variance analysis we have to perform, and then we have to go for the uh, finding out the best model for that particular species. Um, yeah. So suppose I just put it here the some of the result from the E. coli, and what kind of the observation I got, what kind of the output we got from there, and acetic acid formula and all those. I'm just putting here some of the you can say the result which I obtained from the metabolic mediated network mediated process, which is I have done in the Cobra, one of the Python or MATLAB based approach. Basically, you can see these are the different, you can say, metabolites is produced by these E. coli, like acetic acid, ethanol, formic acid, hydrogen indole acid, and lactic acid, succinate acid. And then once we done the experiment and we try to find out whether such kind of the output are coming, flex balance are coming, it's again I will tell you the flex balance method, or you can say these computationally is quite intensive, and how we have to get it, how we have to feed that mechanism. Like it's like a biochemical reaction is going from one end to another end, and what kind of the output is coming, what kind of the biomass is coming from that reaction. So we are able to basically able to find out like 39% of the acetate are producing, succinate are producing, and again it is producing urea is also producing, glycerol is also. So these kind of the things are produced by that bacteria. Some of them like D-methanol is also there, where you can see the D 0% are there. It has to be produced by this lactate also, lactose also, but somehow in my experiment that particular things has been not produced. So anyhow we have to then again now optimize this model and try to like whether we can give more input fluxes. We have to change the fluxes and find out that in large scale how it is going to happen. So suppose that this is one example of E. coli. There are multiple bacteria are there, multiple things are there, and whatever substrates are producing, and the next step is going to how they are interacting with them. Once they are interacting in the different genes, the different proteins, we have to see whether the protein interaction is good over there or not. All those kind of things we have to see. So now I just done this similar kind of exercise in the lactic acid bacteria, which is like more known for the card and all those things, and which is beneficiary for us actually. And then we found like number of total reaction and produce metabolites and all those things are happened over there. There are two different species which are majorly participating in forming of the milk to curd. So one is Delviker cry and H. helvi Yes, another one is this one. So we have find some differences over there. Number of metabolites are produced by both different, and reactions numbers is also different. Um, yeah, of course. Then we go for the flux balance. Again, these programs or these methods are designed in the Python, or some of them are R-based method is also there. But yeah, um, um, Cobra is the main tool which is performing this kind of the analysis. Quite frequent people are using. Again, these are the licensed version. The company those are associated with them, like fermentation industries, are supposed to have this kind of the tools. They are working on that. And then I was discussing about the K-base. This is the entire K-base are designed for the microbial community, and where we can find a lot of related things over there. So I was supposed to give you people the demo about the K-base. I think those are the newcomer here. They can understand about the micro. If they want to understand about the microbiology, basically microbe-based science, they have to definitely explore this particular database, where you can find n number of the things, uh, mm, and the possible integration of the tools, how we can uh, learn this thing. But definitely this tool will not support everything, but for a large amount of the data, amount of the things, we have to do our own things. So this is basically environmental for the system biology. And it has I will go for the it provide the both kind of the system. One is like a developer system. Those want to develop their algorithm and submit there, they can also do. Another kind of that, the automated tools are there. They can use, create and log in and put their data and do the analysis from their system. It's something like Galaxy kind of the things, but it has a bit difference from them. And so so this is the basically an interface where people can log in over there. And after login, uh, will get the we have to log in by you can say the Orchid ID, Google, and all those things. Let me go with, I don't know. No, I think so. I mean to say this: they are having the this kind of the system over. And once you log in with your system, you will get the all different kind of the databases available here, and all the different kind of the tools they are used inside the system. And you can take your data as a input data over here, and then try to analyze that data in a way which we want actually. So those are interested, they can go inside the database and try to explore that in depth. I will come into the another. Yeah. So one of the other database which I want to focus here, which is important, that is like Host-microbe metabolic interesting database, a genome scale studies were happening. So virtual metabolic human is a one of the database which is important database, which contain across the, like you can say, most of the human gut microbes real information. uh, that is about 818 microbes are there. It was like 2018 update, 2019 update, but that database was shifted to Agora, one of the another database which are having the most of the information over there. Agora is containing like more than 7,200 strains and species, which is coming from the human gut microbes. And this has the, you can say this particular database have more precise information, and with the collaboration of the Cobra, and they are having more metabolites over there. Of course, that Agora is consist of the strains across the different species, different host, not species, different host, and uh, you can see here the figure how the profiling of the all the metabolites are happening, what kind of the sample we can collect fecal sample, plasma sample, serum sample, and other type of the health kind of the meta omics data we can generate. And then we can go for the taxonomic and functional profiling of the gut microbes. That is very important. If you are not going, if you are not classifying the different gut microbes across the taxonomy, that is very important. Then we have to go for the reconstruction of microbial uh, GEM actually, and then you can say construction again, as I told earlier, like genome-wide genome-wide mediated metabolic network reconstruction using your Cobra and other tools. And then once we have this kind of the construction, we can find out like how different you can say how the metabolites are interacting with the human gut, or any kind of the interaction is happening, and host how it is actually behaving actually. So this is like overall, like I just given metabolite mediated network based analysis we can perform. Again, it's not a simple task, it's not you can say one day task to happen. It will take at least days, at least 1 month time task to understand the things, how it is happening, how the data is coming, how the we are predicting the pathway, how we are simulating the things, how they are interacting to the host, that's coming output coming, which are coming from the different microbes, as well as from the different foods we are taking, how the system is behaving. So that all these things we have to understand, and yeah, some of the part of things we can simulate in the computational based approaches actually. And coming to, I think then we'll go for the like some of the enlightening. I just put it there, the protein interaction methods, or protein metabolites interaction methods, which is basically used to like it's a very known methods. These are the known methods, but now we are implementing into the you can say microbial study, microbial host studies. So like again, in the first figure I showed, there are many receptors are there, and many flagellum are there. They are interacting with the human body. So how they are interacting, how strong interactions are there, and how they are helping to get attached or get insight into the body and replicate them inside into the cell actually. So I just put here the basics, like what are the different protein interactions approaches are there. One approach is called like interlock approach. I think figure is saying how they are interlocking to with each other. Another approach is domain interaction approach. Is with respect to protein domain are interacting, and if any of the protein are having such kind of the domain, they will interact with the host protein. Then 3D structural approach, entire protein to entire protein approach are interacting. Then we will go for the gene behavior hood approach, which is happening in genome wide approach. This can be things which happening in the genome wide approach, not in the protein level. And gene cluster approach is also we can use if say similar kind of the gene are falling in the same cluster. We can say yeah, these are the particular genes that can be go and bind to the certain receptors of the. Then we can go for the phylogenetic profiling and literature mining based approach also. So these kind of the method we can use for the identifying the protein interaction. It's a general method. It is widely used in human case, human microbe interaction as well as human or any kind of the other species interaction with the things. Next, I just want to explain here about different approaches which are people now using, basically AI based or ML based approach. Docking based approach, I think people are here. I don't know the people how much they have gone through, but I will tell you these are the docking based approach. 3D structure docking based approach is the well known approaches are there. And I just focus like Megadog, JackDog, and LightDog are the or somehow old tool. Those are doing protein index approaches. Deep Rank is the one of the latest tool which is developed in Python and using the ML based approach to do the protein interaction, especially taking the 3D structure. And then once we go for the sequence based approach, we are having deep learning approaches used here. We'll have a string of the sequences, classifying them, generate the features from there. After generating the features, we'll try to the method will try to give you some scores of the interaction. Major methods used here is like sprint, one of the best method for the proteome, human proteome. And descript is the one of the another method which is well known, not well known but recently evolved in a such a way that people are using because the AI it's used the ML based approaches. And again, deep deep trio is also one of the tool, and PEP4 is also their cross species, and deep script will use for the interspecies. Uh, and then another method is there like machine learning based approaches based on the available data sets. So this method is basically if you have some certain known microbes are there and they are having known sequences with us, and we also know the host proteins, those are interactive with them, so we can develop a model. So basically this is like a ML based model has been developed for that. Recently this kind of the work is happened in the case of our tool has been developed in the case of COVID, because we have a certain set of virus proteins. You can say virus protein is there, and known function. We know the known function is there, and what kind of the all proteins are interacting with the human host. We are already known, so all known information are there. We have taken out the feature from there. After taking out the feature, we have modeled them. If some kind of the similar kind of the proteins are coming, we can predict what this particular sequence can be bound to this particular host protein. So this kind of the things can be happen using this machine learning approach. Another things like we can go for the protein interaction network analysis also. This is like we have to we are having a lot of databases which is like providing you the one protein. If you have what kind of the networks are connected with this particular protein, most of the tools are there like Interactome, Intact, Sting is well known, MNIT, PHI based, BioGrid, and Fisto is also there. Those are like basically used to identify the or provide you the entire network information of that particular protein. I did some. This is like a 3D structure based approach. Again, I think I have explained it, but again, I'm just putting in the picture over here, which is taking host is there, bacteria is coming, flagellant of the bacteria is going inside the body, and they are interacting with them. So I just put it somehow like TRL2 receptor, TLR5 receptor, and TLR4 receptor is there. And you can say different probiotics proteins are pleptologite, flagellant, and these are interacting with them. In that way, I think we can find out the interaction. Just give you one of the known and good tool in case of the human I just want to provide this information because that tool is quite updated and quite known. We called as HMI PRED, host microbe interaction prediction, which is basically based on the 3D structure based interaction approaches. You have proteins from the bacteria or probiotics or fungi or viruses. Give the name of that particular protein or put your protein in their interface. They will provide you the entire human proteome interaction. What are the uh, number of protein, your input number of protein of that particular bacteria, where the possibly they are interacting to the different proteins available there. And they will also provide the rank whether it is harmful or it is activating the protein, what it is, or inhibiting the protein. Those kind of fascinated information they will provide in one go. And in case of microbial, that kind of the you can say workflow is also there, how they are actually doing actually. So it will take microbial protein, as I again explain, I'm again explaining that, and then it go and attach to the surface of the different protein which are available, and then it will find out the potential HMIs, protein host microbe interaction. And then if it is favoring in the energy bonding energy are finding in the proper way, they will give you the output for that. I just put it some of the result from this particular database. uh, the microbial protein, you can see here the protein ID are there, and it is interacting to the model organism, which kind of the protein name is also given, and which kind of the human protein it is interacting. So whatever the human protein names are there, it has been put it over there, and their protein name, and then their ICSC score, respective ICSC score. So we can at least see this particular protein of the virus or of the bacteria, what kind of the things can be happen. So if you see the case number, uh, bile receptor is of human protein is uh, mm, basically consuming the butyric acid and producing the butyric acid from the bacteria. Those are interacting with them actually. So this kind of the receptor we can identify and find out the interaction mechanism. After finding out the interaction mechanism, we can come out with some conclusion. Yeah, these bacteria may harmful or may cause something effect because they are interacting with such kind of the protein inside the human body. So this is the one of the good example over there. uh, I think I just put it there. I don't know with this much is this information is required for you people. uh, yep. That is like deep mining approach, deep learning approach, the tool which are taking, how they are generating. Basically, this is here. I just want to present the concept of sequence-based approach. How they are extracting the feature. After extracting the feature, how they are going to the predict that interaction actually. So suppose in figure A, you can see protein 1 and protein 2 is there, and their respective sequences are there, and they have a fixed length. We have to this fixed length are there. Then we have to design the profile of the protein or peptides. And then we once we have the protein, then there are different AI based modules are there where we can rank them accordingly. After ranking them, we can generate the mm, a model which is having a specific kind of the accuracy. So if they are having this a certain accuracy, we can take them to predict the another type of the interaction. So this kind of the approaches people are using nowadays. Yeah. So I just go on one by one. I can go in depth of each of them. Machine learning based like sequence pickup approaches is also there. Then we have. If I just want to explain this protein interaction network concept also here. So we have another different tools that is called like MNIT, Interact, Sting, and other tools is also there. And one of the well-known tool for the network analysis we know as like cytoscapes, which have all the models from the different bacteria, and where we can find out the interaction between them and identify the network, which particular kind of the metabolites are supporting there or participating in there. If you are interested in some certain metabolites, and this metabolites how it is affecting to the different proteins or protein entire protein networks. So in such a study we can go and do that. Again, these are some of the databases which are especially designed for the pathogenic interaction with the human body, where you can find the most of the information related to that. I will not go into the detail. I think PHI waste is the one of the known database which is having the maximum pathogen. Those are listed till now, and they are having they are providing the precise information of each pathogen on how they are affecting, what kind of the mechanism they are using, and how they are interacting with them. So there are like 264 pathogen information are there, and 185 host interaction data is there actually. So like there are and they also cover different like 462 disease they are covering actually. So we'll get what kind of the disease are affecting, which bacteria affecting this particular disease actually. So this is like a summary kind of the pathogenic database which we are having. FIS2 is quite old database, but yeah, updated one is like pathogen PHI based. Still, because in case of bacteria we have to find out the like in case of microbe we do not have you can say the 3D structure, more 3D established 3D structure available into the PDB actually. So definitely we have to go for the modeling approach, and after doing the modeling we have to validate them and all those things. On the other hand, we have to also think about that whether we can avoid those process and we can use some AI based machine learning based approaches to reduce that effort actually. So nowadays people are working in that direction, and they are using the sequence based approaches and trying to get these kind of the protein interactions. Yeah, I think I have concluded there.